Hi everyone, welcome to the session. This is the second lecture of MD 6028 Virology. What we're going to talk about today is viral classification. So in the first lecture we talked about what viruses are. So we introduced some of these key concepts. We looked at the key definitions of what viruses are. So we looked at this in some different ways. We answered this question from some different perspectives, but hopefully through that lecture you now have a really clear idea of what viruses are. Again, you will have had some understanding of this from previous modules. What I wanted to do was make sure everyone's kind of to the same level with regards to this understanding. So moving into the module, we're all comfortable knowing exactly what we're talking about when we talk about viruses. And as I said, today we're going to talk about virus classification. So viruses, as you may be aware, are different to the other groups of microorganisms that we talk about in microbiology. They're actually very, very different. They have some really unique properties, and that's what we're going to talk about today. We're going to talk about how we can classify viruses because they're so different to these other groups of microorganisms that we can't necessarily always classify them in the same way. Okay, so this is what the lecture is going to look like in terms of structure. So we're going to start off talking about some ideas around virus classification, talk about some of the problems that we can face when trying to classify viruses, talk about exactly why we have to approach virus classification differently to how we approach classification of the other groups of microorganisms. And then we're going to actually look in some detail at the different classification groups, or the different classification strategies, I should say, that we can use to classify viruses. So we're going to look at phenotypic classification, we're going to look at phylogenetic classification, and we're going to look at the Baltimore system of classification. Phenotypic classification and phylogenetic classification, you may be able to, just from the names, figure out the type of classification systems we're talking about. Baltimore classification system, this is something really specific. It's named after a person. So again, don't worry if you're not aware of any of these systems. We're going to talk through them today, and that's what we're going to focus on in the lecture. Okay, so firstly, when we think about classification of organisms, microorganisms or macroorganisms, so any form of life, we tend to think about phylogenetic classification. So when we say one species is related to another species, for example, we're talking about phylogenetic classification. When we talk about the Latin names for different species, these names are based in this phylogenetic classification. What phylogenetic actually means is a relationship based upon genetic similarity. So based upon evolutionary closeness is another way we can think about this. So the closer species are in evolutionary perspectives, the more genetically similar they are. So what we can actually do is we can map out species genomes onto a big tree based upon how similar the genomes are. And what we see is when we have groups or branches of species that have similar genomes, they are evolutionarily related. They belong to a specific group. This is how phylogenetic classification works. It's how we have the groups of species that we're kind of all aware of. So if we're talking about fish, if we're talking about mammals, if we're talking about reptiles, these are all classification groups that are based upon phylogenetics. So clearly really useful classification system that does work. What we find is that we can take all different forms of life and we can map them onto a tree of life. That's where the term comes from, tree of life. And this tree, it's obviously enormous with all different species on, and it branches and branches. And as we've said, the closer species are on this tree, the more genetically similar they are, which means the closer they are in evolutionary terms. And the further away they are, the less close they are. So the more genetically diverse they are, and the further away they are in terms of evolutionary history as well. So, and again, if we look at specific bunches on this tree, specific branches, we see groups of evolutionary relatedness, which again, we can define as mammals or reptiles or amphibians or fish, or any of these other terms that we use to group species phylogenetically. I've got this term on the slide, which is LUCA. LUCA means last universal common ancestor. So one principle of this tree is that there is a common base, because the tree has one trunk, right? So every species that is placed on this tree, whether it's at the end of the branches or wherever it is, ultimately it descends from the same ancestor, and we use the term last universal common ancestor. So this is species that sit right at the base of the tree. Obviously it's long dead in terms of evolutionary history, so it doesn't exist as a distinct species anymore, but the point is that all currently extant, so living species, are evolved descendants of this last universal common ancestor, and that's why all different life forms that we currently know about, at least, can fit onto this single tree of life, because they are all descended from this last universal common ancestor. And this classification system is really, really useful. Partly it's just useful from an academic perspective, just because it is interesting to understand how all different forms of life relate to each other. And partly, if we want to think about infectious agents and disease, it's really, really useful for us to understand where microorganisms that cause disease fit onto this tree of life, because when we group things together in phylogenetic terms, we know they have similar properties. So if we think about an example we've used before, reptiles, if we know something belongs to the reptile group, we know it shares features of the other reptiles. Same for mammals, if something belongs to the group of mammals, we know it shares features with the other mammals. Same for insects. Countless examples. And again, if we go back to the example of microorganisms that cause disease, if we take a microorganism and we know it belongs to this particular group of microorganism, then we know it shares features with those microorganisms, and those features will include how it causes disease, what type of treatment we can use against it. So it's really, really useful for us to know that information. Clearest example of this to demonstrate this point is mycobacteria, so tuberculosis. So we can group bacteria in one portion of the tree, so they fit in this one big branch. And then what we can further do is group within this group. So we have groups of bacteria that are similar to each other within this big group of bacteria. Right, so we can split up all bacteria into groups of similarity, and one group is the mycobacteria group. So mycobacteria are a family of bacteria, and it includes TB. And because TB is in that group, or Mycobacterium tuberculosis, the bacterium that causes TB, is in that group, we know it shares similarity to other bacteria in that group. One of the other bacteria within that group is Mycobacterium leprae, so it's the mycobacterium that causes leprosy. So leprosy and tuberculosis seem like very, very different diseases, but you can actually treat them with the same drugs, sometimes very similar drugs, but we call them antimycobacterials, and these are drugs that work against the two. So clearly we can get really useful information from these groupings. If we want a more general example, so a little bit less specific, we can even just think about bacteria as a whole. So bacteria are a group on this evolutionary tree of life, and we know that because bacteria are grouped together, they share some features, and that's why we can treat bacteria with antibiotics, because antibiotics target some of these key characteristics that they share. But if we try and target microorganisms that aren't bacteria with antibiotics, so if we try to target viruses with antibiotics, it just doesn't work. So again, just a clear example. And the same is for eukaryotes. So if we try and target parasitic worms, which are eukaryotic, causative agents of disease with antibiotics, they're not going to work, because they don't have those shared characteristics. They're not bacteria, they don't belong to that group. And the opposite is true. If we try and treat bacterial infections with antiparasitics, it's not going to work. If we try and treat antibiotic infections with antifungals, or antifungals with antibiotics, it's just not going to work. So again, even at the most basic level, this classification system is really, really useful, because it straight away starts giving us information on the causative agent of disease that we're thinking about. Okay, so these are terms that we're almost certainly familiar with on the right: species, genus, family, order, class, phylum, kingdom, domain. So these are the names for the groupings that we have on the tree of life. So these are the levels, if you like. Levels of relatedness is another way of thinking about it. So species is the name of the individual species. Genus is a group of species, so a small branch on the tree of life. Family is a group of genuses, so it's a bigger branch. Order is a group of families. Class is a grouping together of orders. So as we move through these classifications, we're getting closer and closer to the base of the tree of life. And ultimately, right at the base, we have three domains. We have archaea, bacteria, and eukarya. And these domains ultimately, all three of them branched off from the last universal common ancestor. So we want to think of this big branching tree of life on which all forms of life fit, and at the base of this tree of life we have the last universal common ancestor. And if you're looking up at individual species, you'll see they can be classified using this system, no problem at all. So just some examples here. You can look up any species on Wikipedia, and you see the kingdom, phylum, class, order, family, genus. So all forms of life fit into these categorizations. And again, really useful for us to have this understanding. Giraffe here, the class is Mammalia, so we know it shares features with other mammals. So again, it's a really useful classification system. And the example we talked about earlier, Mycobacterium tuberculosis, we know it belongs to the domain bacteria, so we know things about it already. We know it belongs to the genus Mycobacterium. So again, more useful information there, as we were talking about earlier, in terms of treatment. So what we tend to do when we find a microorganism that we're not 100% sure, maybe what it is, how is it related to anything else, what we do is we genetically analyze it. We sequence its genome, then we compare that sequenced genome to the genomes of all other microorganisms that we know about. So we can then, essentially, we're doing phylogenetic analysis, so we can say what other microorganisms it is most similar to, and then we can then hopefully say, okay, it fits in this family, or it fits in this genus, and this gives us really useful information about the microorganism. Okay, so now what we want to do is bring this back to viruses and virology. So I've told you all these rules about classification, which you're probably familiar with from earlier biology modules. The problem is that none of this really applies for viruses. So we've said all different forms of life fit on this tree of life. Viruses don't, and again, it comes back to this key issue of a way and whether viruses are alive or not, because they're so different to other forms of life. So every other form of life, if you sequence its genome, you can fit it into a branch on this tree of life. So you can assign it a place. You can see how similar it is to other forms of life. You can see the path by which it evolved, really. Again, this isn't true for viruses. So when you sequence viral genomes and try and fit them on the tree of life, you see they just don't fit anywhere. They just don't fit. What you actually see is, even when you sequence all the viral genomes and you try and look how viruses are related to each other, you can't answer that question either. They don't even fit in that way. So not only do viruses not fit on this tree of life, but they don't even appear to fit on their own tree of life. The best organization efforts have ended up with at least 7 different branches of viruses. So what actually happens is you can organize the different viruses into some groups, so about 7 different orders. If you remember, an order is one of the levels of classification, but we can't determine how those orders are related to each other. And what's more, most viruses don't even fit into these established orders. So we can essentially say viruses don't fit onto the tree of life. Some viruses can be organized into 7 different trees of life, so 7 groups of relatedness, but we don't know how these 7 groups are related to each other. We don't know when they evolved or where they came from, and most viruses don't even fit onto these 7 trees of life. So really, really complicated, really, really confusing. It doesn't appear that they descended from Luca. So we've said all forms of life on the tree of life descended from the last universal common ancestor. Doesn't appear to be true for viruses. Again, we're just not sure where or when they evolved, and we're not sure about that evolutionary history, and the best we can do is organize them into these 7 groups. You see this term on the slide, which is polyphyletic. So when we think about organisms on the tree of life, so every other form of life on Earth, it's monophyletic. So it's evolved once, if you like. You can see the root, it's split here, and then it's evolved here, and then it's ended up with a single species. Okay, this isn't true for viruses, so it appears viruses have arisen independently multiple times through evolutionary history. So if we look at this evolutionary, or these branches of the evolutionary tree on the right of the slide, we can see again our example of reptiles. Reptiles have arisen once. That means all reptiles are related to each other. This doesn't appear to be true for viruses. It appears they're probably polyphyletic, poly meaning many. So they have many different evolutionary origins, and again, this is just one of the factors that's making it really, really complicated to find out how viruses are related to each other. There's other reasons why it's really difficult to find out how viruses are related to each other. They have really rapid evolutionary rate, which we'll talk about later in the module. They have a tendency to steal genes from host cells and donate genes, which can make it even more difficult to track their evolutionary history, because you can't track a consistent change in their genome because they're randomly picking up genes and losing genes. And again, the best we can really do is group them into a number of different orders. So we can't put viruses into classes or phylums or kingdoms or domains. It doesn't work. We can just put some of the viruses into these different groups, into these classifications, but we don't know how these classifications relate to each other. We don't know where most viruses that are sitting outside of these orders fit either. So we have these groups of classification, these orders, don't know how they're related to each other, and most viruses don't fit into them. And this is why if you go and look up the classification of viruses, just like we did earlier for some non-virus species, you'll see that for viruses, it's a little bit different. You don't get an answer above order. So let's have a look for HIV here on the left. There's a genus there, there's a family and a subfamily, and there's an order, but there's nothing further up. There's no domain, there's no kingdom. Same for Ebola virus. So we can see there's a genus, we can see there's a family, we can see there's an order, but no kingdom, no phylum, no domain. Same for hepatitis B virus. Species, genus, family, order, and then the classification system that we've talked about, phylogenetics, is abandoned because these viruses just don't fit into a class, they don't fit into a phylum, they don't fit into a kingdom, and they don't fit into a domain. What you can see instead is something called a group, and this group refers to the Baltimore classification system that we're going to talk about later in the lecture. So again, phylogenetic classification breaks down for these viruses. It doesn't work for viruses past order, so we use different classification systems instead, and the classification system that happens to be shown on Wikipedia is the Baltimore classification system, and again, this is one of the classification systems we'll explore today. Okay, and just some details of the classification of an example virus here. We have human immunodeficiency virus, and what we see is human immunodeficiency virus is within the family of Retroviridae, and it's with a specific genus, so it's within the lentivirus genus, and it sits in this genus along with a number of other species of viruses. Okay, so we have these HIV-1, HIV-2, and SIV, simian immunodeficiency virus. These are lentiviruses. So lentivirus is a genus, and these are the genuses as well. And they all fit into the family retrovirus. And I want to reinforce the point that this classification is really, really useful information. So earlier we talked about some examples of why classification is useful in terms of therapeutics and microorganisms that cause disease for bacteria and fungi and things like that. And the same is absolutely true for HIV and all other viruses, because when we group viruses together, even though we don't know the context of these groups, so we don't necessarily know how these families of viruses related to other families, we don't know how the orders are related to the other orders, it's still really useful to group viruses together because they have these shared features. And an example of the usefulness of these shared features that I talked about earlier was the idea of therapeutics. So if you can put a microorganism into a particular group, you can say, oh, we can probably treat it in a similar way to other members of this group when it's causing an infection. Yep, absolutely true. But we have another example here, which is these retroviruses have a tendency to cause cancers. You can see from the names of the species here, sarcoma virus, leukosis, myeloblastosis, mammary tumor virus, leukemia virus, sarcoma virus, leukemia virus, lymphotropic virus, which causes leukemia as well, hyperplasia. So these retroviruses all have a tendency to cause human cancers. Obviously, this is really important. We care about viruses that cause human cancers, and we know that the ability of retroviruses to cause human cancers is linked to the way that they replicate, and they all replicate in a similar way because they belong to this group. They are evolutionarily similar, so they have evolved this very similar strategy to replicate within cells. And this strategy is inherently linked to their ability to cause cancers. So again, it's really, really useful for us to know that these viruses are related to each other because it gives us a lot of information about how they are causing cancer. Okay, with viruses, we tend to think of families as being the most useful distinction. So as we've said, most viruses don't fit into these orders. We only have about 7 orders, and most viruses sit outside them. So it's not super useful to think about orders, just because most viruses don't fit in them. But what we do tend to think about is families, because most viruses will fit into families. Again, we don't necessarily know how these families relate to each other, but still family is a useful classification in and of itself, as we showed in the last slide. So it's useful for us to group viruses into these little families because therefore we know a lot of information about the viruses in the families. We know about their shared features and how they replicate and cause disease and things like that. So we very frequently use virus families when we're talking about viruses. Okay, so those are some of the issues we face when we're trying to classify viruses. We've also talked about why it's important to classify viruses. Hopefully, we're clear on that point because it gives us useful information. Again, with another example with retroviruses, we use antiretroviral drugs to treat retroviruses. So we have drugs that specifically target the processes that retroviruses undertake. So therefore it's useful for us to group these viruses together because we know we can treat them in this way. Okay, so what we want to do now is look at some of the other classification systems that we can use for viruses. As we've said, there are some real issues with classifying viruses in the traditional way. So what we want to do is think about phenotypic classification as an alternative. We want to explore phylogenetic classification a little bit more because, as we've said, it's a really, really flawed classification system, but it's still useful to a degree. And then we want to talk about Baltimore classification system as a really interesting way that we can classify viruses. Okay, so the first type of classification system we want to talk about here is phenotypic classification. Now, again, based on the name phenotypic, you may be able to guess what the classification system is broadly based on. Essentially, it's based on the phenotype, and what we mean by phenotype is observable characteristics, so outward characteristics, right? If we think about viruses, we can think about some specific outward characteristics that they potentially have, and we can use these characteristics to classify the viruses. This is what we have demonstrated on the slide. We have the key features that we can use to classify viruses phenotypically. So the key phenotypes that viruses can have is another way we can think about that. Firstly, we can use the genome of the virus. So as we've talked about, a virus is just a genome wrapped in protein, sometimes wrapped in a lipid envelope. So straight away we're almost limited in terms of the phenotypes we can observe because we're talking about something that is so simplistic and has so few components. However, again, we know all viruses have a genome, so we can use this genome. We can see what the characteristics of this genome are and use this to classify the viruses phenotypically. You're probably used to genomes being double-stranded DNA, particularly if you've been studying cell biology. Of course, you've been talking a lot about double-stranded DNA. Viruses do not necessarily have double-stranded DNA genomes. They do not necessarily have DNA genomes. They have genomes that come in a range of different forms. So different viruses have different types of genome. Again, this is why it's useful to classify viruses based upon their genome type, because we can see different viruses have a different type of genome. And what we can also see is the type of genome they have impacts the way they replicate, and it can impact the way they infect the host, and it can impact a lot of other things as well. It can impact which therapeutic drugs we can use to target a particular virus. So this phenotypic classification, hopefully you're beginning to see, can be really, really useful, and that's one of the reasons why we use it. So back to the viral genomes, as we've said, the viral genome is not necessarily double-stranded DNA. The viral genome can be either DNA or RNA. It can be double-stranded or single-stranded. So we have double-stranded DNA genomes and single-stranded DNA genomes. And we also have double-stranded RNA genomes and single-stranded RNA genomes. So straight away, there's a range of different genome types there. We can also think about whether the genome is positive or negative sense, and we'll talk a bit more about what this means later on. But essentially, when a virus has a single-stranded RNA genome, that single strand can be positive sense or negative sense. And this is a really useful characteristic for us to understand and for us to use in classification, because again, it directly impacts how the virus replicates, and that in turn impacts a whole bunch of things. So the type of cell it can infect and the type of therapeutic agent we can use against it. So again, a really useful part of this classification. Finally, we have the envelope. So the first type of classification in phenotypic classification is based upon the genome. The second feature we can use is the envelope. So we said some viruses have an envelope and some don't. We can use this as part of phenotypic classification. So we can essentially say, is the virus enveloped? Does it have this lipid envelope, or is it naked? Is it unenveloped? And these are the two categories within the envelope portion of this classification. So overall, phenotypic classification is actually quite simple. We're just looking at the type of genome, and then we're also looking at whether or not the virus has an envelope. And again, the envelope classification, or the envelope feature used as classification, is really, really useful because it impacts how viruses interact with the cell, how they replicate, and impacts what therapeutic agents we can use. So you may be aware of a type of antiviral called fusion inhibitors. If you're not already, you'll definitely be aware of them by the end of the module. Fusion inhibitors inhibit the process by which enveloped viruses enter into the cell, specifically enveloped viruses, because the lipid envelope, when the enveloped viruses enter into the cell, actually fuses with the host cell membrane, and fusion inhibitors inhibit this process. Obviously, it's for viruses unenveloped. If it doesn't have a lipid envelope, then there's no fusion. So a fusion inhibitor will not work against unenveloped viruses. So again, really, really useful classification system. These tend to be the two features of a good classification system. One, does it actually work? Can it be used to classify what we want to classify? And the problem that we said with phylogenetic classification is it doesn't really work all the time. A lot of viruses can't be classified phylogenetically beyond family, and none can be classified beyond order. For phenotypic classification, however, it does work. All viruses can be classed phenotypically. All viruses have a genome, and all viruses are either enveloped or unenveloped, so they can be classified using the system. And the other feature of a good classification system is whether or not it's useful. Can we actually obtain useful information from this classification? We've said for phylogenetic classification, we can obtain useful information for viruses. So when we can classify viruses phylogenetically into the little families and sometimes into orders, then it's really useful to do that. However, phenotypic classification is also useful because again, we're getting this useful information about how the virus replicates, about what potential therapeutic agents we could be thinking about using. And this is what it looks like when we actually apply this classification system. So if we take viruses and we apply this phenotypic classification, this is what we see. First off, we tend to apply the genome portion of the system. So we split viruses into DNA viruses and RNA viruses. Then we further split the viruses into single-stranded and double-stranded. So is their genome a double-stranded genome, DS, or is it a single-stranded genome, SS? For RNA viruses, we can then split the single-stranded viruses into the positive sense genome viruses and the negative sense genome viruses. So we can say, do these viruses that have single-stranded RNA have a positive sense single-stranded RNA or a negative sense single-stranded RNA? And again, we'll talk later about what this actually means, but the key point is it is useful classification. It gives us information about the virus, and also it works. So all these single-stranded RNA viruses can be described as positive sense genomed or negative sense genomed. So it's really useful and it works. Finally, we can ask whether the virus, well, any particular virus we're trying to classify is enveloped or unenveloped. And as you can see, this is the lowest level on the classification here. And then that's the classification system applied essentially. And then what we do is we put families onto this classification tree. So as we've mentioned, we can use phylogenetic analysis to group viruses into families, and it's really useful to do that. However, the problem is then how do these families relate to each other? Difficult to know through phylogenetics. So what we can actually do is kind of put these families together based upon phenotypic classification. So the names on the bottom here, these are phylogenetic families, and we're using the phenotypic classification system to group these phylogenetic families into other groups. Okay, so we're grouping phylogenetic families together. So all families that have a DNA virus, that have a DNA genome, and that genome is double-stranded and enveloped, they will be grouped in the bottom left here. So Herpetoviridae is a family with these features, Herpes viridae is a family with these features, and Pox viridae is a family with these features. So what we actually tend to do is combine phenotypic classification with phylogenetic classification. And another way we can think of that is we take these phylogenetic families, so these families that we've obtained through phylogenetic classification, and we group these families based upon phenotypic classification. So hopefully that makes sense. So what we'll do now is look at some of the aspects of this classification system in a little bit more detail. So firstly, as we said, we can use the genome. So we can classify the viruses as either being RNA or DNA. And by that we mean they have an RNA genome or a DNA genome. We can also say the genome is either double-stranded or single-stranded. And again, this is a really useful way to classify viruses. One reason is because DNA viruses tend to have some different properties to RNA viruses. Because if we can say a virus has an RNA genome, we can already imply some features of that virus. One big feature is the mutation rate. So RNA viruses tend to have a much more rapid mutation rate than DNA viruses. The reason for this is DNA viruses tend to be double-stranded, as you can see from the diagram. And you can also see that RNA viruses tend to be single-stranded. Now, if a genome is double-stranded, so you have two strands, if there's a mistake in one of the strands, that mistake is noticed by error. There's proofreading mechanisms to detect errors. Basically, if there's a mistake in one strand, there's a mismatch between the two strands, and there's mechanisms to detect this, so that's repaired. For the RNA viruses, however, because they only have a single strand, if there's a mistake somewhere, so the wrong base is incorporated, there's no real way for the virus to detect that. There's no proofreading mechanisms in the cell that the virus is infecting that will detect that. So what you end up with is RNA virus genomes accumulating a large number of mutations. And again, this has consequences downstream. It has consequences for how the virus actually survives in a host. Its ability to accumulate these mutations can be problematic for hosts because it's difficult for a host to deal with a virus that replicates and mutates very, very quickly. And again, we'll explore this in more detail, but again, the principle is this classification is useful and it matters. And the reason why DNA viruses do tend to be double-stranded is simply because DNA is most stable when it's double-stranded. And for RNA viruses, we can think of the opposite as being true. So RNA is most stable when it's single-stranded. And this is why RNA viruses tend to be single-stranded. All this goes some way to explaining why a lot of the emerging viruses, so a lot of the viruses that seem to have suddenly evolved an ability to infect a new host, so to infect humans, tend to be RNA viruses. And again, this is partially linked to their fast mutation rate. Okay, and again, like a lot of these rules, it's not absolute. There are some double-stranded RNA viruses. Virology is a family of double-stranded RNA viruses, and we also have single-stranded DNA viruses, and parvoviride is the principal example we use there. Okay, next we also said that RNA virus genomes that are single-stranded are also categorized into either being positive sense or negative sense. So what we want to do now is explore what we actually mean by this. So to understand this distinction between positive and negative sense nucleic acid, we can look at double-stranded DNA in host cells, so in a eukaryotic cell. And as I'm sure we know, our genomes are in the form of double-stranded DNA, and this DNA has obviously two strands, is double-stranded. One of the strands is negative sense, one is positive sense. What this means is one of the strands is directly used as the base sequence to produce transcripts, which will then be translated. So the exact sequence in this strand is what determines the amino acids in the ultimate protein. The other strand of DNA, however, is just complementary to that strand. So we have the encoding strand and we have the template strand. The encoding strand is a strand that actually contains the information. It actually contains the sequence that will determine the amino acid sequence. The template strand is just complementary to this, so it just base pairs it to it. And again, one of the reasons is because of the way DNA is replicated, this double-stranded structure allows the cell mechanisms to maintain the integrity of the sequence overall. So really important we understand we have the positive strand and the negative strand. Positive is encoding, negative is template. So when we think about RNA viruses, as we've said, most RNA viruses are single-stranded. obviously a single strand can't be positive sense and negative sense. It can't be a coding strand, an encoding strand, and a template strand. So RNA viruses that are single stranded, their genome will either be encoding or template. So it will either be positive sense or negative sense, and this has really significant implications on the way the virus replicates inside the cell. Because if the viral genome is positive sense, so if it's an encoding strand, then that genome can essentially just be translated straight into protein. Okay, so from your lectures around cell biology, you should understand that in a normal host cell, the process of protein expression means going from double-stranded DNA, using that double-stranded DNA to produce mRNA, and then this mRNA is translated into protein. Now, we've said some RNA viruses will have a positive sense genome, and their genome is RNA. So essentially, it is no different from mRNA in the cell. It is no different in kind of structure to the mRNA that is in the cell. So positive sense RNA genomes of viruses can just be directly translated into proteins when the virus enters into the host cell. For RNA viruses with negative sense genomes, however, this doesn't apply, this can't occur, because as we've said, negative sense genomes don't directly contain the information. They first need to be used to produce a positive sense RNA, and then this positive sense RNA can be translated into protein. And this is the big difference between positive sense and negative sense RNA viruses. For positive sense RNA viruses, their genome can be directly translated as soon as they come into the cell, because their genome is essentially mRNA, so it's treated like mRNA by the cell machinery. It is translated into protein. For negative sense RNA viruses, as we've said, a negative sense strand of RNA doesn't contain the information that's needed to produce a protein. It contains the complementary information. So the only way that a negative sense RNA virus can produce protein from its genome is to first produce a positive sense copy of its negative sense genome. So use its negative sense RNA as a template to produce an encoding strand. And then this encoding strand is then translated into protein in the cell. It's a really tricky concept. Again, we're going to go into it in a lot more detail as we go into the lecture, as we go into the module. The main thing to remember at this point is the key definitions and the general ideas I'm putting across, and also why it's useful for us to use this classification system. And again, it's useful because these two types of virus are replicating in a very different way, because their genomes are different, because a positive sense genome and a negative sense genome can't be expressed in the same way, because there's this additional step that negative sense RNA genomes need to undergo. Okay, so the next stage of this classification is whether the virus is enveloped or unenveloped. So as we've said, viruses can either have an envelope or can not have an envelope, and this envelope that we're referring to is just a lipid layer on the outside. So again, viruses can be enveloped or unenveloped, and this is one of the ways we can classify them phenotypically. Okay, so as we've said, enveloped or unenveloped, this is true for DNA viruses and RNA viruses. So RNA or DNA viruses can be enveloped or unenveloped. Again, it's really useful for us to use this type of classification because it impacts how viruses enter into the cell, so it impacts how they replicate, and again, this in turn determines how we can intervene therapeutically. So there are specific drugs, fusion inhibitors, for example, that will only work against enveloped viruses. They won't work against unenveloped viruses, so therefore it's important that we understand the difference between the two, and we understand which key viruses belong to which group. Okay, and that's just a run through of phenotypic classification. So hopefully it's been clear, hopefully it's been useful. Again, everything on this diagram has been explained. We have the key ways that we can classify viruses phenotypically. We have discussed the basis of these different ways we can classify, so the types of different genome. And we've also touched on the idea that some viruses have a lipid envelope and some don't. We've also reinforced the idea that this classification is useful. It's important. It gives us useful information about the viruses. Okay, so we discussed at the start how there are real issues with phylogenetic classification of viruses, but we've also said we do still use it to a degree. So we still classify viruses phylogenetically into families, because that is quite a useful thing to do. However, when we try to classify viruses further up, so beyond that, so try and find out how these families are related to each other, we run into quite a lot of problems. So if we've said a lot of viruses will fit into orders, but most won't. So yeah, it gets quite complicated, but again, we do use phylogenetic classification to a point. We do classify viruses phylogenetically into families. And again, as we've said, this it does work to a point. So it works into families, then we run into problems. You'll see these suffixes used, virus for genus, virinae for subfamily, viridae for family, and virals for order. So this viridae is the important one, because when we're talking about virus families, we'll see the suffix is viridae, and when you see that suffix viridae, it means we're talking about a family. And we've already been using this suffix because we've already been using the terminology relating to virus families. So we've talked about retroviridae, we've talked about parvoviridae, reoviridae, poxviridae. Clearly you can see the pattern. Suffix is viridae because we're talking about virus families. So we say here 9 orders, 7 orders. If we're saying 9 orders, we're including bacteriophages as well. So bacteriophages it's a distinct order of viruses. Again, depending upon the literature, you'll see 7 or 9. So either is fine. The point is that there's a limited number of orders of viruses, and that number is more than 1. So we have these multiple trees that viruses fit into. But again, we don't know how these trees relate to each other, and most viruses don't fit into these trees. Okay, so these are the orders that we do have. Again, it's not something you really need to memorize. The only point I'm making here is you can see the suffix virals, which means we are talking about orders. You'll also note if you look in the literature that these change around fairly frequently. Again, one of the issues with virus classification is it's difficult. A lot of things aren't 100% clear. So if you look at the literature, you'll see a lot of back and forth about the details. Okay, one point that I do want to make, so an additional point here, is that if we try to look at these orders and compare them to phenotypic classification, we see there's a big overlap. So for herpes virals, we're talking about a lot of double-stranded DNA viruses that are enveloped. And we can see some more examples of this here. So the point that's being made here is that there is this overlap, and that actually makes sense because when we're talking about phylogenetic orders, we're talking about viruses that have genetic similarity, so they have a shared evolutionary history, and therefore it is understandable that they have similar phenotypic characteristics, right? If we're talking about a group of viruses that are genetically similar, so they've evolved together, it makes sense that they have the same genotype. Also makes sense that they share other phenotypic characteristics, like whether or not they have an envelope. And again here, just reinforcing that same information. So when we look at the order, we see the viruses within the order fit some particular characteristics. So they all fit in a similar place on the phenotypic system. But again, as we've said, most viruses don't fit into this order. So viruses that do fit into this order share these traits, but not all viruses that share these traits fit into the order. So hopefully this makes sense. It's just an elaboration of the idea that this phylogenetic system, this order-based system, excludes most viruses. Okay, and here's a paper I just want to draw your attention to. So it's a fairly recent paper in which the authors tried to put together a system for classifying viruses. So again, as we've said, there are these massive problems with classifying viruses phylogenetically. Every now and again, a paper will come along trying to impose a bit more order on the systems that are in place. Really interesting to read them, but none of them really address the key problems, because the key problems can't be addressed. The key problems that viruses are, polyphyletic, that viruses don't necessarily have a shared evolutionary history, and that we don't understand how viruses relate to other forms of life. So this is an interesting paper. It's just proposing a slightly different structure for how we classify viruses phylogenetically, but the key issues are still there, really. Okay, that's phylogenetic classification. Again, just reiterating the point that we do use phylogenetic classification for viruses up to a point, but again, at that point, we face these really significant issues that, at least for now, are insurmountable, and we can't successfully classify all viruses phylogenetically. However, we can group them into families, we can make some attempt to put them into these different orders, and it is useful for us to do that. So again, we do it to a point. Okay, so finally we want to talk about Baltimore classification system. So this is the final classification system we'll talk about. This is a classification system that was actually created specifically to classify viruses. So again, as we've talked, there's issues with the other classification systems, and Baltimore classification was essentially created as an updated version of classification for viruses. Okay, so we talked about if we look up viruses on, just for example, Wikipedia, because this is what people do if they first want to, learn some information about a virus. The first place to go is usually Wikipedia, and if we do that, we can see the attempts to classify these viruses phylogenetically. We can see how it goes up to family and sometimes order. So we have an order for HIV and Ebola virus, but for HBV, it's unassigned. And an interesting point here is we're talking about quite important viruses sometimes. So hepatitis B virus is a virus that has a significant impact on human health globally. However, it's unassigned. So don't slip into the thinking that, okay, we managed to assign the important viruses into orders. We understand these important viruses that we've studied in detail and that we've researched. It's just the other viruses that we don't understand as well. It's not the case. We have these really important viruses that we don't necessarily understand how they relate to other viruses. And again, that we've established that with phylogenetics, and we've said that if we look at these classifications, there's this classification that appears above order, called a group, and this group shouldn't be familiar to us. So the other terms on the right here, species, genus, family, order, and all the rest to domain, we're probably familiar with from our studies of biology and just from, generally hearing these terms being used in the context of zoology. So we probably know just animals and other forms of life belong to a family and an order and a kingdom and a phylum. However, group is not a distinction that's used in phylogenetics, really. It's used generally to say, oh, this is a group, but it's not an actual classification level on the phylogenetic system. As we can see on the right, there's just no group there. We just have species, genus, and all the rest, but group is not a distinct level of classification. Group on the left slide, so group being used to denote viruses, is actually in reference to the Baltimore classification system. So it is laid out a little bit confusingly on Wikipedia actually, which isn't great because it looks like group is the next level up from order in phylogenetic classification system, but that's not the case. That's not the case at all. Up to order, we're talking about phylogenetic classification. Then for group, we're talking about something else. We're talking about the Baltimore classification system. Again, slightly confusing how it's laid out on Wikipedia, but that's just the way it is. Okay, again, these groups are not phylogenetic. These groups are based upon the Baltimore classification system, and that's what we're going to introduce now in the final portion of the lecture. Okay, and again, Baltimore classification system is the classification system we're going to discuss now. So it's an alternative to the other classification systems we've talked about. However, as we've seen previously, there is overlap. So, for example, we saw there's overlap between phenotypic classification and phylogenetic classification, which means that whether or not we classify viruses based upon their phenotype, or we try to classify them based upon their phylogenetics into these orders, we see there's overlap. So viruses in the same order tend to be placed in the same position on the phenotypic classification system, and this is just because viruses that have evolved together, that have these shared evolutionary history, they tend to have the same phenotype as well. And again, we see that with the Baltimore system. We see viruses that have evolved, have a shared evolutionary history, I should say, tend to have the same phenotype, and they also tend to be placed together on the Baltimore classification system. The reason is the Baltimore classification system is based specifically upon the way viruses transcribe and translate their genome. So the way viruses express their genetic information into protein, and obviously this is quite closely linked to certain features of phenotypic classification, and it's also quite closely linked to phylogenetic classification, because viruses that have a close evolutionary history tend to also express their genome in a similar way. So we're seeing consistencies across the three, which is absolutely fine and it's absolutely expected, but it's just something we want to be aware of. Okay, so this is Baltimore classification. This is the best graphical representation of the system. As you can see, there's 7 different classifications there. So there's 7 different groups, and all viruses will fit into one of these groups. So again, it's a useful classification system because it includes all viruses, so it works, it just works. We can classify all viruses using it. It's also a useful classification system because it gives us information about viruses, so it gives us useful information. So whichever group a virus gets assigned to, we obtain really useful information again about how it expresses its genome. Okay, as we said, 7 groups. You will see the top level of each group. We have terms denoting genomic material. So in the dark green boxes, double-stranded DNA, single-stranded DNA, double-stranded RNA, and then the 2 types of single-stranded RNA. And then we have ssRNA-RT, and then we have dsDNA-RT. What this refers to is the type of genetic material that the particular virus has. So in group 1, the first group, viruses have double-stranded DNA. Obviously, as we've said, there's a huge overlap here on phenotypic classification system, because we know in phenotypic classification system there is one of the basis of phenotypic classification is the type of genome that the virus has. So clearly already we're seeing this huge overlap. However, the distinction between phenotypic classification and Baltimore classification system is that Baltimore classification isn't just dependent upon the type of genome the virus has, it's actually dependent upon the way viruses express their genome. Because what we'll see is some viruses, even though they have the same type of genome as another virus, they fit into different groups because they express the genome in a different way. So most double-stranded DNA viruses will fit into group 1 because most double-stranded DNA viruses express their genome in a particular way, but some double-stranded DNA viruses will fit into group 7, so the group at the very end, because they express their genome in a slightly different way. So this is the distinction between the Baltimore classification system and phenotypic classification system. Okay, you'll see that there's this arrow beneath each of the boxes This arrow is basically signifying that the virus is going from its genome to RNA. Okay, so every virus expresses its genome via RNA. The reason is that every virus uses the host cell translation machinery. So every virus in a host cell undergoes the process where ultimately RNA is translated into protein. And again, that's a host cell process, because in every host cell, RNA is translated into protein. That's what translation is. So therefore, every virus must undergo the same process, because it's using host cell machinery. So every virus must have as an endpoint of expressing the genome this RNA that can then be translated into protein. So we can split genome expression into two stages, transcription and translation, as we know. Translation is turning RNA into protein, so that's the second stage. The first stage is going from genome to RNA. So genome is used to produce RNA, this is transcription, and then the RNA is translated into protein. This is what happens in a host cell, and this is the process that viruses must participate in as well. And again, because viruses are always using host cell machinery to translate their genome, they need to be producing RNA, because in a host cell, the process of translation goes from RNA to protein. So these are really complicated concepts now that we're going into, and we're going to explore them in a lot more detail in later lectures, but I hope at least the key points I'm making are clear. And the key point here is that the Baltimore classification system is dependent, it classifies viruses based upon how they produce RNA, because all viruses must produce RNA when they're in their host cell, because that's how they can express their genome. Okay, so as we can see, the line for each group is going from the original genome to RNA. Because again, as we said, they all need to produce RNA. So for group 1 viruses, these are double-stranded DNA viruses, and they produce mRNA from their genome. Single-stranded DNA viruses, they produce RNA from their genome, but do it in a different way. Double-stranded RNA viruses, again, produce RNA viruses from their genome, but do it in a different way. Class 4, positive single-stranded RNA. Class 5, negative single-stranded RNA. Class 6, single-stranded RNA RT. RT stands for reverse transcriptase, so we're going to talk about this in later lectures. Essentially, group 6 viruses are single-stranded RNA viruses that have reverse transcriptase, so they use reverse transcriptase to produce RNA. And group 7 viruses, these have double-stranded DNA genomes, but they use reverse transcriptase to express that genome, so to produce the RNA. And again, as we've said, there's a significant overlap between Baltimore classification and phenotypic classification, because Baltimore classification is dependent upon how viruses express their genome, and how viruses express their genome is very closely linked to the type of genome that a virus has. And hopefully that's clear from the slide, so we can see how these groups overlap significantly. Back to these Wikipedia diagrams that we see. So just to clarify what we're seeing when we look at the group information here. So we have Ebola virus. The group here says group 5, and in brackets negative single-stranded RNA. So it is a group 5 virus on the Baltimore classification system, which if you look back at the diagram we just looked at, means it is a negative single-stranded RNA virus, and it expresses its genome just like the other negative single-stranded RNA viruses. So we also have hepatitis C virus, and hepatitis C virus, as we can see, is denoted as being group 4. So group 4 means it's a positive sense single-stranded RNA genome that the virus has, and this genome is expressed like other positive single-stranded RNA genomes. And again, we have the phenotypic classification system on the right, and on this classification system I've overlapped the different groups or the different classifications from the Baltimore classification system, and we can see there's this really significant overlap. We can see group 1, 2, 3, 4 and 5 have clear positions on the phenotypic classification system. Group 6 and 7, this is where things get a little bit more complicated, because as we've said, these are viruses that have types of genome that are represented in the other groups. However, they express the genome in a different way. And this is why Baltimore classification is in many ways preferable to phenotypic classification, because it has these extra groups. It allows us to understand that some viruses are different to others despite having the same type of genome and despite having the same envelope status, whether they have one or not. Okay. So a lot of technical information there in the Baltimore classification system. The good news is we're going to go over that in a lot of detail. It's really interesting, really important that we understand how these different viruses express their genome, and that's what we're going to talk about in some subsequent lectures. So the main takeaway from this Baltimore classification part is that Baltimore classification is based upon how viruses express their genome, and again we're going to explore that. We're going to go group by group and look at exactly how that occurs. Right, okay, thank you very much everyone for your time, and I'll see you in the tutorial when we work through these concepts a bit more.